Welcome back to my channel. Today we will be starting a new topic in biochemistry, that is glycosaminoglycans and mucopolysaccharides. Okay, so we'll just start with it. Now, if you talk about what do you mean by glycosaminoglycans or mucopolysaccharides, first it is very important that it is a long unbranched heteropolysaccharide with a repeating disaccharide unit. So definition, so remember glycosaminoglycans means what? It's a long, so it is long, but then it is unbranched and what? Heteropolysaccharide and has got repeated disaccharide unit. What is this repeated disaccharide unit, sir? Very important. Disaccharide unit consists of two things: one, amino sugar, and then acidic sweet sugar. Okay, now what does amino sugar contains? Listen carefully. It contains glucosamine as well as galactosamine. Okay, and then we have acidic sugar remaining, that is the glucuronic acid. It contains glucuronic acid as well as hyaluronic acid. Understand? Okay, so the definition is it's long, unbranched, and has got repeated disaccharide units, and also it is a heteropolysaccharide molecule. Understand? Okay, now what are the properties of the glycosaminoglycans? Very important, sir. Listen carefully. First, it contains large number of negative charges. Now you can ask me a question: why contains negative charges? Very important. See, we already learned it is a disaccharide unit. It contains amino sugar as well as acidic sugar. This acidic sugar, we know it is a negative charge, yes or no? Acidic sugar means what, amma? Negative charge. Okay, so but then this amino sugar is there, no? It is positive. Then how, sir, it is negative charge? Listen carefully. Along with amino sugar, it also contains sulfur, 2 minus sulfur, as well as CH3CO minus. So in all these things, in all these compounds, since it is attached with the amino sugar, it makes the glycosaminoglycans more negative. Understand? So it contains large number of negative charge. And also in nature it is very slippery. Why, sir? See, why it is slippery? Due to the repelling structure or nature of the all negative charges. So when we have a lot of negative charges coming together, it repels. That makes nature it is slippery. Okay, very simple, right? And also the joints are mobile. Why, sir? Because of presence of glycosaminoglycans in the synovial fluid. Okay, so in synovial fluid we have the glycosaminoglycans. We'll learn later. Okay, so in the synovial fluid we are having the glycosaminoglycans, and that makes the joints more mobile. But then listen carefully, the joints become stiff on aging. Why it becomes stiff on aging? Because the concentration, the content of the glycosaminoglycans will decrease on aging. That's very important. Okay, because of the decreased content of the glycosaminoglycans. Very important, very simple, right? Okay, now next point is it binds to large amount of water. Okay, large amount of water, and thus this glycosaminoglycan is a very important component of extracellular matrix. It's a major component of the extracellular matrix. Now we have one component which is intracellular matrix. It acts on, it is the only glycosaminoglycans which is present in the intracellular. Tell me what is that? We'll learn later. Okay, okay. Now what do you mean by mucopolysaccharide? It is the time to know about what do you mean by mucopolysaccharide. Now when the glycosaminoglycans is present in the mucin, okay, it is present in the mucin, then we call it as mucopolysaccharide, which means muco means mucin. Understand? Remember it in this way. So mucin, where the glycosaminoglycans is present in the mucin, then we call it as mucopolysaccharide. And very important characteristics, that is the glycosaminoglycan is resilient. That you have to know. Now it is time to know about what is the major glycosaminoglycan. First important thing, we will learn the names first. Okay, we have the hyaluronic acid, chondroitin sulfate, keratin sulfate. Okay, then heparin sulfate, and then dermatin sulfate. Now let us learn about the disaccharide units. So we have learned about the disaccharide units, what all is there: amino sugar and then acidic sugar, and we have seen the units. Right, now for hyaluronic acid, when you learn all those disaccharide units, okay, in exam point of view, if they ask for 5 mark question, it will be very confusing which is for N-acetyl glucosamine, which is for N-acetyl galactosamine. Okay, so if you learn everything in order, it will be very confusing when you write for the examination. Now how to learn, sir? It is important. See, first you take only one disaccharide unit. Okay, you learn only the one pattern of disaccharide units. That is, for hyaluronic acid, we are taking as N-acetyl glucosamine. Okay, then search for any other N-acetyl glucosamine is there. That is Keratin Sulfate. Okay, so N-acetyl glucosamine is present in the Keratin Sulfate as well as hyaluronic acid. But then in hyaluronic acid, it contains glucuronic acid. Okay, N-acetyl glucosamine plus glucuronic acid. So where it is present in the skin, synovial fluid, bone, cartilage as well as the loose connective tissue. Okay, so N-acetyl glucosamine plus glucuronic acid present in the hyaluronic acid. Now what is this Keratin sulfate? N-acetyl glucosamine, but then it contains galactose. You understand? Very simple. Okay. Next we will learn about the next disaccharide unit, that is the N-acetyl galactosamine, where it is present chondroitin sulfate. Okay, N-acetyl galactosamine, and also we have here, that is the N-acetyl galactosamine, where dermatan sulfate. Okay, so in the chondroitin sulfate we are having N-acetyl galactosamine, and also it contains of glucuronic acid. Very simple, right? Okay, and but here dermatan sulfate contains a very important acid that is called as hyaluronic acid. That is the dermatan sulfate. So here it is glucuronic acid and N-acetyl galactosamine in the chondroitin sulfate. Dermatan sulfate contains hyaluronic acid plus the N-acetyl galactosamine. Okay, next important thing is heparin. So there is no acetyl group of the either glucosamine or galactosamine, but it is only glucosamine, heparin plus hyaluronic acid. Okay, plus the hyaluronic acid. Heparin sulfate is what, that is glucosamine plus glucuronic acid. So this is how you have to learn about the major things. Okay, this is the about the disaccharide units. Okay, it's very simple. Okay, now we will be learning about the characteristics feature of all the chondroitin sulfate. What is that, First thing about chondroitin sulfate is the most abundant, and also it will be present mostly in the cartilage. Okay, so it is the most abundant and major component of the cartilage. And what is the function in cartilage, Major function is for compressibility of the cartilage, right? Understand? So this is for the compressibility of the cartilage. What about keratin sulfate? Keratin sulfate we have a different type, that is case 1, keratin sulfate 1 and keratin sulfate 2. Okay, now this keratin sulfate 1 very important, present in the cornea, so only responsible for the transparency of the cornea. Okay, this for the transparency for the cornea, and also it is mostly heterogeneous and contains no uronic acid. Very important, one more question, no uronic acid. Okay, that is the keratin sulfate. Okay, but keratin sulfate contains what? Very important, that is N-acetyl, we have learned here. So N-acetyl glucosamine plus the galactose, so only contains no uronic acid. Understand? Okay, next we learn about the heparin sulfate. Now this heparin sulfate is mainly to be acting as a plasma receptor. So plasma membrane receptor, that is the heparin sulfate. What it does? Very important. See, in the vascular endothelium, it contains LDL. Okay, to hold this LDL in the vascular endothelium, we are having this heparin sulfate. So it anchors the LPL, that is the lipoprotein lipase in the vascular endothelium. Okay, understand? In the endothelium, that is vascular endothelium, we are having more amount of LDL, but to hold this LDL, what, who is there? Heparin sulfate. Understand? And also in the kidney, in the basement membrane, if you see, okay, we this basement membrane, it is purely negative. Why? It contains glycosaminoglycan and mainly the heparin sulfate, that is responsible for the negative charge in the kidney that is in the basement membrane and also charge selectiveness. So if you think about basement membrane for the kidney is more negative, then it's due to the glycosaminoglycan, that is the heparin sulfate. Easy. Now what is the function of heparin? So we know that heparin is purely anticoagulant. Okay. Now I ask the question from here, which is the intracellular glycosaminoglycan. We have the answer, that is the heparin. So this is the intracellular, only intracellular glycosaminoglycan present in the where in muscles, skin, lung as well as liver. Very simple. So only intracellular glycosaminoglycan, I am repeating so much, that is the heparin, and it is an anticoagulant, you all know. Right. So when you inject this heparin, very important, when you inject this heparin, it is going to release the LPL from the vascular endothelium. It is going to release the LDL from the vascular endothelium. So who is holding the LDL from the vascular endothelium? Heparin sulfate. To release the LDL from the vascular endothelium, it is heparin. Very important. Okay. Oh. Now dermatan sulfate, you already know, it is present in the dermis, that is the skin. Okay, and it is widely distributed. Fine. And also it is responsible for the structure of the sclera. Structure of the sclera. And very important, related to the LDL, that is it binds to LDL forming atherogenic. Okay, forming the atherogenic component. Fine. Next, very important, that is the hyaluronic acid. So hyaluronic acid contains no sulfate group and it is covalently attached to the, and is not covalently attached to the protein because it contains no sulfate group. Okay, so only when there is presence of sulfate group we can attach to the protein, but then no sulfate group, so we cannot attach to the protein. That is the hyaluronic acid. What is the function? It is important for the cell migration. So cell migration is very important for what? Wound healing, tumor metastasis. Okay, tumor metastasis and also morphogenesis. So it is responsible for the muscle migration. That is, we having the hyaluronic acid. Understand? Now when you compare the proteoglycan as well as glycoprotein, what is the function? It differs from the percentage of the carbohydrate. Yes, for both it is present of carbohydrate is there, but how much percentage? So for proteoglycan it is more than 5% and for glycoprotein is less than 5%. Now what is the type of carbohydrate present in between two? Very important. Proteoglycan we have the glycosaminoglycan, and for glycoprotein is purely oligosaccharide. Do proteoglycan contain repeated units? Yes, because it is glycosaminoglycan, the presence is there. So we are having the repeated units, disaccharide repeated units. Okay, but since it contains oligosaccharide, there is no repeating units. This you have to know. Now what is the structure of proteoglycan? Very simple, listen carefully. So the main starch is the protein. So we are having the starch as protein. Okay, and in that starch we are having lot of glycosaminoglycans. We are having lot of glycosaminoglycans. Now you tell me, who is going to anchor or hold the glycosaminoglycan? Okay, someone has to be there to anchor or hold the glycosaminoglycan. That is the function of trisaccharide unit. Trisaccharide units. So what are those trisaccharide units? That is the galactose, xylose. Okay, we are having here. Okay, so galactose, xylose and galactose. So trisaccharide unit contains of galactose and xylose. Very simple. Okay, so the starch is protein and the trisaccharide unit, that is this units attaches the glycosaminoglycan to the protein. We have learnt. So for the synthesis of the glycosaminoglycan, we need that is present in the endoplasmic reticulum. But degradation of glycosaminoglycan occurs only in the lysosomes. Okay, this we learn about the glycosaminoglycans. In part 2, we will be learning about the mucopolysaccharides. Thank you.